You might think you know the basics of where human life begins. But what if the very first chapter of your existence was far more dramatic, more consequential than you ever imagined?
Hmm.
It's this period of incredible complexity, mostly unseen, yet it profoundly shapes who we become.
It truly is. Today, we're taking a deep dive into that extraordinary journey of prenatal development and the birth process itself.
Yeah.
And this isn't just about the biology, which is amazing in itself. It's really about understanding that foundational first chapter of human life, how it shapes every aspect of us.
And the huge questions it raises, right? Legal, ethical,
Right.
social.
Exactly, all that. So our mission today is to kind of unpack this intricate period. We want to give you some surprising facts, some crucial insights.
The goal really is for you to walk away not just informed, but genuinely fascinated by this hidden world before birth.
Definitely.
Yeah.
And to really underscore just how critical and complex this development can be, maybe we should start with a couple real-life cases.
Good idea. Like the conjoined twins, Gracie and Rosie, born in England back in 2000.
Right. They shared vital organs. I believe it was one heart and a pair of lungs between them. Biologically, Rosie was completely dependent on Gracie for survival.
Which presented just an agonizing dilemma for everyone involved.
Absolutely. Doctors knew that without separation, both girls would inevitably die. But separating them meant Rosie wouldn't survive, though Gracie might have a chance.
And their parents, devout Catholics, felt it should be left in God's hands.
Understandably. But the surgeons, they felt this moral obligation to save one life if possible. So they took this incredibly difficult decision to the High Court.
That court intervention was really remarkable, wasn't it?
It was. The arguments were intense, comparing it to a mountain climber cutting a rope, calling them two persons in one body, both with a right to live.
Yeah. And ultimately, the court ordered the separation.
They did. Rosie Thadley died, as predicted, but Gracie survived. And she's actually a healthy young woman now.
Amazing. And these aren't isolated cases, are they?
No, not at all. South Africa had its own very high-profile example. Mimfo and Memfonyana, born in '88, connected by their heads, sharing brain tissue.
I remember that case. They were separated.
Yes, successfully separated at 17 months.
Mhm.
Though sadly, Mfonyana died 2 years later. Mfo, the surviving twin, she lived until 2021, I think, to the age of 34.
Wow.
Yeah, and she experienced only relatively mild brain damage and some paralysis on one side, hemiplegia.
So these stories, right from the start, they just instantly illustrate the profound effect prenatal development has on postnatal life.
Exactly. And it's funny because for centuries, developmental psychologists didn't really give the prenatal period much thought, psychologically speaking.
Right. It was seen as just biological prep work.
Pretty much. It really wasn't until maybe the 18th century that we even fully grasped that life truly began as individual cells.
But today, that view is completely different.
Oh, completely transformed. Researchers now recognize the immense impact of this period, and it's largely for two main reasons, I'd say.
Okay, what are they?
First, decades of research have just consistently shown that numerous prenatal and birth-related factors significantly affect later psychological development, like really significantly.
Okay, that makes sense. And the second?
The second reason, well, it comes back to cases like the conjoined twins. It became undeniable that these early stages don't just affect individuals. They profoundly impact society legally, ethically, religiously.
It forces us to confront really deep questions.
Absolutely. It's like trying to understand a whole book without reading the first chapter. You're missing the foundation for everything that follows.
That's a great analogy.
And this whole area, it also ties deeply into behavior genetics or psychogenetics, exploring the genetic basis of our behaviors.
Okay, so that's a perfect jumping off point then. To really understand this foundational chapter, we have to start right at the beginning. The spark of life, conception.
Right, that incredible moment. It all begins at a fraction of a second, usually in one of the fallopian tubes, when a single sperm meets an egg cell.
And that union, that's conception, or fertilization.
Exactly. Conception, fertilization, same thing. And that newly formed single L, that's the zygote.
And the scale of this, it's pretty mind-blowing, isn't it?
It really is. Egg cells, the ova, they're the largest cells in the human body, but still barely visible. A female is born with, what, about 400,000 of them?
But only a fraction are ever released.
A tiny fraction. Only about 300 to 400 will ever be ovulated during her entire reproductive lifetime. It's incredible.
Contrast that with sperm cells. Tiny, right.
Among the smallest cells. And males produce an astonishing number maybe 300 million sperm daily.
Daily. Wow. Yeah.
And while a typical ejaculation might release, say, 100 to 500 million sperm, you only need one, just one for fertilization.
And it's an epic journey for that one sperm.
Oh, absolutely. Navigating the acidic environment of the vagina, getting through the cervix, reaching the fallopian tubes. Millions perish along the way. It's quite the gauntlet.
So it really is a race against the clock, too.
Definitely. Sperm can survive for up to 5 days inside the female reproductive tract. But the egg cell, it's only viable for fertilization for about 24 hours after ovulation, usually less, maybe 12 to 36 hours.
So a very narrow window.
Very narrow. And once that one winning sperm penetrates the egg, something amazing happens. The egg's outer wall instantly changes, basically sealing it off. No other sperm can get in.
Wow. And in that moment?
In that precise moment, the nuclei of the sperm and egg fuse. And boom, you have that new cell, the zygote, containing all 46 chromosomes, 23 from the mother, 23 from the father.
But, and this is maybe surprising to some, not every fertilized egg actually develops into a baby, does it?
No, not at all. It's estimated that about 25%, a quarter, are spontaneously aborted in the first few weeks. Often so early, the woman doesn't even realize she was pregnant.
And then even later?
Right. Another 30% or so miscarry later in the first trimester. The reality is, fewer than half of all fertilized eggs actually result in a living baby.
That's quite sobering.
It is. And just zooming out for a second, it's interesting to note global birth rates are generally declining, especially in industrialized countries. Often below that replacement rate of about 2 children per woman.
But not everywhere.
No, definitely not. In contrast, some sub-Saharan African countries still see birth rates over 5 births per woman. In South Africa, where about 1 million babies are born annually, there's often a correlation seen between lower education, lower income, and higher birth rates. It's complex.
So back to that single cell, the zygote. It holds the blueprint for an entire human being. How?
It starts with the chromosomes, right?
Exactly. Those 46 chromosomes arranged in 23 pairs, one set from each parent. And these chromosomes are essentially packed structures made of segments called genes.
Genes, the smallest units of heredity, carrying all our characteristics.
That's right. And the estimate now is that humans have somewhere around 20,000 to 25,000 genes.
Which is less than we used to think.
Mm-hmm.
Yeah, quite a bit less than earlier estimates, which were closer to 100,000. There's still some uncertainty because of different detection methods and, genes can overlap in functions.
Yeah, we don't even know what most of them do yet.
That's the truly remarkable part. The functions of most genes are still unknown. We're still figuring out how they interact with each other, how they interact with the environment. It's incredibly complex.
Okay, and this is the part that always fascinates me. The actual chemical basis of it all. DNA.
Ah, DNA. Deoxyribonucleic acid. The recipe, the blueprint for life. It's this huge, elegant molecule, that famous double spiral, like a twisted rope ladder.
And the truly astounding thing isn't just how it works, but how similar we all are, genetically speaking.
It's mind-boggling, isn't it? All humans are something like 99.9% identical in their genetic makeup. That tiny 0.1% difference accounts for all the incredible diversity we see, physical differences, psychological differences, everything.
It's incredible efficiency.
Absolutely.
Mm-hmm.
And DNA strands themselves are unbelievably small. You could fit, 5 million of them through the eye of a needle?
Amazing. But DNA holds the blueprint. It doesn't actually build things directly.
Right. For that, DNA relies on another nucleic acid, RNA, ribonucleic acid. Think of RNA, specifically messenger RNA, as the courier. It takes the blueprint information from the DNA in the cell nucleus out into the cytoplasm.
Where the building happens.
Exactly. It directs the construction of proteins, which are the basic building blocks of pretty much everything in the body.
And mapping all of this out, understanding this blueprint, that was the goal of the Human Genome Project, right?
Precisely. One of the most ambitious and important research projects ever undertaken.
How would you explain its significance, like in simple terms?
Okay, think of the human genome, all our genetic material, as a massive recipe book.
Right.
Your 23 pairs of chromosomes are like the 46 chapters.
Mm-hmm.
And those 20,000 to 25,000 genes, they're the individual recipes within those chapters. The Human Genome Project started in the U.S. in 1990, completed around 2003, basically aimed to read that entire recipe book. Not just map it, but understand it. It really shifted medical research, didn't it? Moving focus from just treating symptoms to understanding the root genetic causes of diseases.
So what were the big results?
Well, huge breakthroughs, a much clearer picture of how DNA duplicates, how genes switch on and off, where they begin and end. It confirmed we have fewer genes than we thought.
Okay.
Crucially, they discovered almost 2,000 illness genes. This dramatically sped up diagnosis for many genetic conditions from potentially years down to days.
That's life-changing.
Absolutely. And it launched whole new fields like pharmacogenetics, studying how your specific genes affect how you react to medications. This leads to more personalized, effective treatments.
But the project being completed doesn't mean we know everything now.
Oh, not by a long shot. It's an ongoing journey. We still don't fully understand the genetics behind complex conditions like heart disease, for example. But the HGP was, and still is, this incredible stimulus for future research. It gave us the foundation.
So we have the blueprint. How does the body actually replicate these instructions to build a person from that single zygote?
Through cell division, right?
Exactly. Two main types. First, there's mitosis. This is for your regular body cells or somatic cells, starting with the zygote. The goal here is simple duplication.
One cell becomes two identical cells.
Precisely, each with a full set of 46 chromosomes. Mitosis involves several phases, but the end result is two genetically identical daughter cells. This process just repeats and repeats, building all your tissues and organs.
Okay, so that's for building the body. What's the other type?
That's meiosis. This is special division just for the germ cells, the cells that become eggs and sperm. Meiosis is about reduction.
Reduction. How so? It reduces the chromosome sets from 2 down to 1. So it takes a cell with 46 chromosomes and produces cells with only 23. This happens in 2 stages.
And crucially, these resulting cells, the eggs or sperm, they aren't necessarily identical to the original germ cell.
That's the key point.
Yep.
They're not identical. Meiosis introduces genetic variation, which is fundamental for evolution and diversity.
And part of how that variation happens is through something called crossover.
Yes. Crossover is fascinating.
Yeah.
It happens during the first stage of meiosis. Imagine the chromosomes you got from your mom and the ones from your dad pairing up.
Okay.
During crossover, these paired chromosomes literally exchange segments. Bits of genetic material swap places between the maternal and paternal chromosomes.
So it shuffles the genetic deck even further.
Exactly. It significantly enhances the uniqueness of each egg or sperm cell's genetic makeup. It's a major reason why siblings, even from the same parents, can be so different. It increases the variation within the gene pool.
Which brings us to how specific traits actually get expressed. You mentioned alleles.
Right. An allele is basically a gene pair responsible for a particular characteristic. You get one allele for that trait from your mother, one from your father, on that chromosome pair.
And if those two alleles are identical,
Then you're homozygous for that trait, and the characteristic will definitely show up. Easy enough.
but what if they're different if you get different instructions?
That's being heterozygous. And this is where dominant and recessive genes come in. A dominant gene's instruction will always win out. Its characteristic will be manifested.
Even if the other allele says something different.
Correct. The recessive gene's characteristic won't be shown in that case. But, and this is important, that recessive gene doesn't disappear. It remains part of your underlying genetic makeup, your genotype, and you can still pass it on to your children.
Okay, can you give an example?
Sure, the classic one is hair color. Let's simplify it.
Yeah.
Genes for dark hair are generally dominant. Blonde hair genes are recessive.
So if you get a dark hair gene from one parent and a blonde from the other,
You'll have dark hair because dark is dominant, but you still carry that recessive blonde gene.
which means two dark-haired people could have a blonde child.
Exactly. If both parents happen to carry that recessive blonde allele and both pass it on to the child will be homozygous for blonde hair and will be blonde.
But a blonde person can't carry a hidden gene for dark hair.
Correct. Because if they had the dominant dark hair gene, they wouldn't be blonde in the first place.
But it's usually more complex than just one gene, right? You mentioned polygenic inheritance earlier.
Yes, absolutely. That's a crucial point. Many normal characteristics like eye color, height, skin color, even things like intelligence and temperament, which we once thought might be simple dominant recessive, are now understood to be polygenic.
Meaning multiple genes are involved.
Multiple genes interacting with each other and often with the environment too. So while the dominant recessive concept is fundamental, the reality for most traits is much more complex. It's rarely just one switch. It's more like an entire orchestra playing together.
And what about diseases and disorders linked to genes?
Interestingly, most serious inherited diseases like PKU, sickle cell anemia, or hemophilia are actually caused by recessive alleles.
Why is that?
Well, think about it. If a serious disorder was caused by a dominant gene, individuals who inherit it would likely be severely affected, often before they can reproduce. So the harmful dominant allele tends to get eliminated from the gene pool relatively quickly.
But there are exceptions.
There are. The classic exception is Huntington's disease. It's caused by a dominant gene, but its devastating symptoms, personality changes, mood swings, involuntary movements, typically don't appear until a person is in their 30s or 40s.
After they might have already had children.
Exactly. So they can unknowingly pass the dominant gene onto the next generation before they even know they have the disease.
Okay, so we've covered how traits are passed down. What about the fundamental trait of being male or female? How is sex determined?
That's determined by our sex chromosomes, which are the 23rd pair. Females have two large X chromosomes, XX. Males have one X and one smaller Y chromosome, XY.
And it's the father's contribution that decides it, right? That's often misunderstood.
Yes, that's a common misconception. It's absolutely the father's sperm that determines the child's sex. Remember, the mother's egg always contains an X chromosome.
Right.
If the sperm that fertilizes the egg carries an X chromosome, the resulting zygote is XX, a girl. If the sperm carries a Y chromosome, the zygote is XY, a boy.
And since men produce roughly equal numbers of X and Y sperm, the chances are about 50-50.
Theoretically, yes. Though, interestingly, slightly more boys are born than girls. In South Africa, the ratio is around 100 to 102 boys for every 100 girls. The reasons for that slight imbalance aren't fully understood.
Now, these sex chromosomes, they don't just determine sex, do they? They carry other genes, too.
Correct. These are called sex-linked characteristics. Genes located on the X or Y chromosome influence other traits, and many of these are more frequent in men.
Why is that?
It comes down to the difference between the X and Y chromosomes. In females, XX, if there's a recessive allele for a condition on one X chromosome, there's often a dominant healthy allele on the other X that can suppress it.
Okay.
But in males,
Yeah.
XY, the Y chromosome is much smaller and carries fewer genes. It often lacks the corresponding allele to override a recessive one on the X chromosome.
So if a male inherits a recessive gene on his X chromosome,
He's more likely to express that trait or condition because there's no dominant counterpart on the Y to mask it.
can you give an example?
Hemophilia is the classic one we mentioned earlier. A mother can be a carrier. She has the recessive gene on one X, but is healthy because of the dominant gene on her other X. But if she passes that X with the recessive gene to her son,
He'll have the condition because his Y chromosome doesn't have the fix.
exactly. That's why it ran through some European royal families, earning it the name royal disease. Other examples are things like red-green color blindness, male pattern baldness, certain types of muscular dystrophy.
And this contributes to what some call a male disadvantage in some aspects.
Yes, statistically, males tend to have higher rates of miscarriage, infant and childhood deaths, more birth defects, and are more prone to certain behavioral, learning, and intellectual disabilities. It's complex, but the genetics of the sex chromosomes play a role.
Okay, so that covers single births. But sometimes development takes a different path right from the start, leading to multiple births.
Right, and twinning rates have actually been increasing, especially in higher-income countries.
Which is interesting, but also a public health issue.
It is, because multiple births, twins, triplets, etc., are associated with higher risks. Lower birth weights, higher rates of prematurity, more complications during pregnancy and birth, and unfortunately higher rates of stillbirth and infant mortality.
What causes these multiple births?
Several factors can increase the likelihood. A family history of twins, older maternal age, certain racial groups have higher rates, even larger maternal size. But the main driver behind the recent increase is definitely the use of infertility treatments.
Makes sense. So what are the basic types of twins?
Two main types. First, monozygotic or identical twins. This happens when a single fertilized egg, a single zygote, splits into two separate entities very early in development.
So they started as one.
Exactly, which means they have the exact same genetic material, they're always the same sex, and typically look very alike.
And the other type?
Dizygotic or non-identical fraternal twins. This happens when the woman releases two separate eggs during ovulation, and each egg is fertilized by a different sperm.
So two separate conceptions happening at the same time.
Pretty much. Genetically, they are no more similar than regular siblings who share about 50% of their genes. They can be the same sex or different sexes.
And this brings us back to those really complex cases, like the conjoined twins we started with, Gracie and Rosie, Mempho and Memphoniana. How do they fit into this?
Conjoined twins are always a type of identical twin. They form when that single zygote starts to split, as if to make identical twins, but the separation process is incomplete.
So the splitting stops partway through.
Essentially, yes. This results in them being physically joined, often at the chest, abdomen, hips, or head. And critically, They frequently share vital organs.
Which leads to those incredibly difficult medical and ethical situations.
Exactly. It's very rare, maybe 1 in 200,000 births. But sadly, a large percentage, maybe 60%, are stillborn or die very shortly after birth. Survival rates for those born alive are also low, perhaps 5 to 25%. And interestingly, they are more often female, about a 3.1 ratio.
It really highlights how complex those very first cell divisions are.
It absolutely does. Tiny changes in that early process can have profound consequences.
Now, for many couples, the journey to even get to that conception stage is difficult, leading to issues of infertility. How is that defined?
Generally, infertility is defined as the inability to conceive after 1 year of regular, unprotected intercourse.
And the pauses are varied.
Very much so. It's roughly split. About a third of cases are primarily due to male factors, like sperm count or motility issues.
Yeah.
Another third are due to female factors, like ovulation problems or blocked fallopian tubes. And the remaining third are due to a combination of both partners' issues, or sometimes the cause remains unknown.
Does age play a big role?
Yes, definitely. Female fertility naturally starts to decline from the mid to late 20s, maybe early 30s, and drops more significantly after 35. The last birth for most women is typically around age 40 or 41.
And for men?
Men continue to produce sperm throughout their lives, but both the quantity and quality tend to decrease after age 40. So paternal age can also be a factor.
Facing infertility must have a huge emotional impact.
Oh, enormous. Couples can experience feelings of anger, frustration, inferiority, depression. It can put a real strain on their relationship, including their sexual relationship. It's a very difficult journey.
But thankfully, there are solutions available now.
Yes, science has made huge strides. Options range from hormone treatments to stimulate ovulation, surgery to correct physical blockages, and of course, assisted reproductive technology, or
RT, that includes IVF.
Yes, in vitro fertilization, IVF, is the most common type of RT. That's where eggs are retrieved from the woman, fertilized with sperm in a laboratory dish in vitro, means in glass, and then the resulting embryo is transferred back into the woman's uterus.
And IVF has become quite common.
Very common. The first test tube baby, Louise Brown, was born in England in 1978. South Africa followed in 1984. Since then, millions of babies have been born worldwide thanks to IVF and other ART techniques.
Is there any discussion about potential risks with ART babies?
Yes, there is ongoing research and discussion. Some large-scale studies have indicated a slightly higher chance of certain health problems or birth defects in babies conceived via RT compared to natural conception.
But it's complicated.
It is. It's hard to disentangle the effects of the RT procedures themselves from the underlying factors that cause the infertility in the first place. The parents might have pre-existing conditions, or older parental age is often involved. So while there might be slightly elevated risks for some things, the overall risk is still relatively low, and millions of healthy babies are born through RT.
And pushing the boundaries even further, there's the concept of cloning.
Right, cloning, which is essentially non-sexual reproduction to create a genetically identical copy of an organism.
Like Dolly the sheep.
Exactly. Dolly, cloned in 1997, was the first mammal cloned from an adult cell. Since then, other animals like mice, pigs, cattle have been cloned.
How does reproductive cloning actually work?
Basically, you take the nucleus, which contains the genetic material, from a regular body cell of the animal you want to clone. You then transfer that nucleus into an egg cell from which the original nucleus has been removed. This reconstructed egg is then stimulated to start dividing as if it had been fertilized and implanted into a surrogate mother.
But human cloning, that's still science fiction, largely.
For now, yes. It's considered much more technically difficult than cloning other mammals. And of course, it raises enormous ethical controversies.
Right. Similar to debates around abortion, I imagine, what are the main arguments for and against?
Well, proponents might argue it could help infertile couples have genetically related offspring, or advance our understanding of cell development, maybe even offer therapeutic advantages like cloning cells to treat diseases like Parkinson's or diabetes by replacing damaged cells. That's therapeutic cloning, slightly different from reproductive cloning.
Okay, and the arguments against?
They are significant. First, the technology is far from perfected. Remember Dolly? She was the one success out of nearly 300 attempts. Cloning has high rates of failure, miscarriages, stillbirths, developmental abnormalities, premature aging.
So the risks are huge.
Huge. And in humans, the potential psychological effects on identity, intellect, emotions are a massive unknown and a major ethical concern. Plus, there are worries about reducing genetic diversity, potential for misuse. Generally, reproductive cloning in humans is widely considered unsafe and ethically unacceptable by the scientific community right now.
So development mostly follows the genetic script, but sometimes errors occur, leading to genetic abnormalities. How common are these?
Thankfully, significant abnormalities are relatively rare. Only about 2 to 3% of live births worldwide involve a major defect.
And these abnormalities fall into different categories.
Broadly, yes. We can differentiate between gene abnormalities, which result from faulty individual genes, and chromosomal abnormalities, which involve errors in the number or structure of the chromosomes themselves.
We talked about hemophilia as a sex-linked gene abnormality. Are there others linked to specific groups?
Yes, there are ethnically linked gene abnormalities. These occur with higher frequency in certain ethnic groups because, historically, an ancestor carried a faulty gene that then became more concentrated within that population over generations through intermarriage.
Can you give some examples?
Sure. In South Africa, for instance, porphyria is more common among Afrikaners. It's a metabolic disorder that can cause severe skin reactions to sunlight. Tay-Sachs disease, a devastating neurological condition usually fatal by age 5, is more prevalent among people of Ashkenazi Jewish descent.
So specific mutations became concentrated.
Exactly. Other examples include albinism, which is more frequent among Black South Africans, or thalassemia, a type of anemia, seen more often in South Africans of Greek or Italian descent. These are typically recessive conditions.
Okay, so those are gene-level issues. What about chromosomal abnormalities?
These involve larger-scale errors, like having an extra chromosome or a missing one. The most common chromosomal abnormality is Down syndrome, also known as trisomy 21.
Trisomy 21, meaning 3 copies of chromosome 21.
Precisely. Instead of the usual pair of chromosome 21, individuals with Down syndrome have 3 copies in every cell, resulting in a total of 47 chromosomes instead of 46.
And what are the typical effects?
There's a wide range, but common characteristics include some level of intellectual disability, certain distinctive facial features like upward slanting eyes, a flatter facial profile, smaller ears, often shorter stature, lower muscle tone. There can also be internal issues like heart defects. Development is usually delayed, and there can be learning difficulties or issues with impulse control.
Is it always the same severity?
No, there's variation, and there's also a rarer form called mosaic Down syndrome, where only some of the person's cells have the extra chromosome 21, while others are normal. This often results in milder features and developmental impacts.
Can these kinds of abnormalities be detected before birth?
Some can, yes. One common prenatal test is amniocentesis. It's usually done between 15 and 20 weeks of pregnancy.
How does that work?
Using ultrasound for guidance, a very fine needle is inserted through the mother's abdomen into the amniotic sac to withdraw a small sample of the amniotic fluid, which contains fetal cells. These cells can then be cultured and their chromosomes analyzed.
But it can't detect everything.
No, definitely not all potential abnormalities can be detected this way, and the test itself carries a small risk. Research is constantly ongoing to improve prenatal screening and diagnosis.
Okay, so genetics plays a huge role, but development is also influenced by the environment the baby is growing in, right? These external factors are called teratogens.
Exactly. A teratogen is basically any external agent that can cause a negative effect on prenatal development. Traditionally, people thought mainly of medical things, certain medications, illicit drugs, maternal illnesses.
But the definition is broader now.
Much broader. It now includes factors like maternal and paternal age, nutrition, exposure to radiation, even the mother's emotional state. The field studying these effects, particularly in behavior, is called behavioral teratology.
So what determines how much damage a teratogen might cause? It's not always straightforward, is it?
No, it's really complex. There are several key principles. First, and perhaps most critical, is the time of exposure.
When it happens matters.
Hugely. The embryonic period, roughly the first 8 weeks when major organs are forming, is generally the time of greatest vulnerability for major structural defects. Exposure later, in the fetal period, might affect growth or brain development, but is less likely to cause gross physical malformations.
Okay, timing is crucial. What else?
Genetic vulnerability. Both the mother's and the developing child's genetic makeup can influence susceptibility. The same exposure might affect one fetus significantly more than another.
Like some people handle substances differently.
Sort of, yes. Then there's the degree or intensity of exposure. Generally, the more exposure, the greater the potential harm. Think dose response.
Makes sense.
Fourth, teratogens often exhibit selective damage. A specific teratogen tends to affect particular parts of the body. For example, The drug thalidomide notoriously caused limb defects, while rubella, German measles, primarily affects the eyes, ears, and heart.
Interesting. So they target specific systems.
Right. And finally, a really important point. The damage isn't always obvious at birth. Some effects, especially behavioral or psychological ones, might only become apparent later in childhood: learning difficulties, attention problems, emotional issues. This is called delayed manifestation.
That's quite sobering, but it's important to remember.
Absolutely. It's crucial to reiterate that abnormal development is the exception, not the rule. Despite all these potential risks, around 97 to 98% of babies born are healthy. The developmental process is remarkably resilient.
Okay, let's dig into some specific teratogens. Parental age, you mentioned.
Yes, there's a general consensus that there's a higher risk for certain problems for mothers who are very young, say under 20, especially under 15 or older, generally considered over 35.
What are the risks for teenage mothers?
They tend to have higher rates of premature births, stillbirths, and complications during pregnancy, like high blood pressure or anemia. This is often linked to socioeconomic factors and biological immaturity as well.
And for older mothers, say over 35 for a first pregnancy or over 40.
They might experience longer or more difficult labors, higher rates of stillbirths, and other complications. And the risk of having a child with Down syndrome does increase significantly with maternal age.
Why is that?
It's thought to be related to the aging of the egg cells. Older eggs are more prone to errors during meiosis, leading to chromosomal abnormalities like the ester chromosome 21.
But it's not a guarantee.
Not at all. It's vital to stress that most women in their late 30s and marrows do not have babies with Down syndrome. The absolute risk is still relatively low for any individual woman, and healthy older mothers often have pregnancies just as smooth as younger women. Some research even suggests the public health focus should perhaps be more on supporting very young mothers.
And what about the father's age? Does that matter?
That's an area of growing research. Historically, it was overlooked. But recent studies suggest that advanced paternal age, often defined as over 40 or 45, might be associated with slightly increased risks for things like fetal death, premature birth, low birth weight, and even certain rare genetic disorders or mental health conditions like schizophrenia or autism spectrum disorder in the offspring.
But the link isn't as strong as with maternal age.
Generally, the risks associated with paternal age seem to be smaller and perhaps less certain than those clearly established for advanced maternal age. But it's increasingly recognized that paternal factors do play a role.
Okay, next up. Maternal nutrition. Hugely important, I imagine.
Absolutely fundamental. The unborn child is completely dependent on the mother for nutrients received via the placenta. A healthy, nutrient-rich diet before and during pregnancy is strongly linked to better fetal health, normal birth weight, and increased survival rates for both mother and baby.
And the opposite, malnutrition.
Malnutrition poses serious risks. This includes both undernutrition, too little food, or an unbalanced diet lacking essential nutrients, and overnutrition.
Let's start with undernutrition. What are the effects?
Undernutrition increases the risk of stillbirths, low birth weight, premature births, physical deformities, stunted growth, and infant death in the first year. Critically, it can have permanent negative effects on the developing brain, affecting size, number of brain cells, overall composition, leading to cognitive impairments, and potentially increasing the risk for mental disorders later in life.
And this is a major issue in some parts of the world.
Yes, sadly. In South Africa, for example, estimates suggest around 30% of the population faces food insecurity. The potential impact of addressing this is massive. Eliminating maternal and early childhood malnutrition could potentially increase average IQ by 10-15 points, cut maternal deaths by a third, reduce infant mortality by over a third, and even boost adult work capacity.
That's staggering. What about overnutrition?
Overnutrition, consuming excessive calories, often from nutrient-poor foods, leads to maternal overweight and obesity. This also increases risks for the baby, including a higher likelihood of obesity and diabetes later in life, and increased risk of infant mortality.
Does the father's weight matter here too?
Interestingly, yes. Research now links paternal obesity before conception to issues like reduced fertility and potential metabolic disturbances in their offspring. So both parents' nutritional status matters.
Okay, what about invisible threats like radiation?
Radiation can be a potent teratogen. The tragic consequences seen after the atomic bomb explosions in Japan during World War II provided stark evidence. Increased rates of physical deformities, later development of heart disease and cancer, growth retardation, intellectual disability, miscarriages, and stillbirths among those exposed in utero.
Does that include medical x-rays?
Yes, X-rays are a form of ionizing radiation. Cells that are rapidly dividing and differentiating, like those in an embryo or fetus, are extremely vulnerable. Levels of radiation that might be harmless to an adult can potentially harm an unborn child.
Is there a particularly risky period?
Yes, the period from about 10 days to 12 weeks after conception is considered the highest risk for major radiation-induced defects. Less severe effects can occur up to about 20 weeks.
Should pregnant women avoid all X-rays then?
Not necessarily. Doctors are very aware of these risks. Necessary X-rays are performed with precautions, like lead shielding over the abdomen. The dose from most diagnostic X-rays is very low, and the risk is generally considered small when weighed against the medical need for the X-ray. Unnecessary exposure, however, should always be avoided.
Moving on to infectious diseases during pregnancy, can viruses and bacteria cross the placenta?
The placenta acts as a pretty good barrier, but yes, some infectious agents are small enough to cross it and infect the fetus.
Like rubella, German measles.
Yes, rubella is a key example. It's usually a mild viral illness in children and adults. But if a pregnant woman contracts it, especially during the first trimester, the virus can cross the placenta and cause congenital rubella syndrome, CRS, in the baby.
And CRS is serious.
Very serious. It can cause a range of problems, including heart defects, deafness, blindness due to cataracts, intellectual disability, liver and spleen damage. It also increases the risk of miscarriage and stillbirth. The risk is highest in the first 3 months, maybe up to 50% if infected then.
How can it be prevented?
The MMR, measles, mumps, rubella vaccine is highly effective. Women should ensure they are immune before getting pregnant. The vaccine itself isn't given during pregnancy. Natural infection also provides lifelong immunity.
What about HIV?
HIV, the virus that causes AIDS, can also be transmitted from mother to child. This can happen during pregnancy, across the placenta, during labor and delivery, through contact with blood and fluids, or after birth, through breastfeeding.
How high is the risk of transmission?
Without any medical intervention, the risk is estimated at 15–45%. However, with effective antiretroviral therapy for the mother during pregnancy and delivery, and for the baby after birth, along with safer delivery practices and alternatives to breastfeeding, the transmission rate can be dramatically reduced to less than 5%, sometimes even below 1%.
That's a huge success story for medicine.
It really is. But without early diagnosis and treatment, about half of HIV-infected infants will die before their second birthday. HIV infection in utero can also be linked to physical abnormalities and neurological problems. Given that around 20% of reproductive-age women in South Africa are HIV positive, prevention of mother-to-child transmission programs are absolutely vital.
Are there other significant infections like STDs?
Yes, definitely. Congenital syphilis, caused by a bacterium transmitted sexually, is a major concern. The bacterium can cross the placenta, especially in the second half of pregnancy, or infect the baby during childbirth.
What are the effects?
Untreated syphilis in pregnancy can lead to miscarriage, stillbirth, or the baby dying shortly after birth. Babies born with congenital syphilis can have severe health problems affecting multiple organ systems, severe anemia, skin rashes, bone damage in large organs, blindness, deafness, intellectual disability. Sadly, rates of congenital syphilis have actually been increasing in South Africa recently.
And herpes.
Congenital herpes, caused by the herpes simplex virus, usually HSV2, is another concern. It's a very common STD. Estimates suggest 40–70% of South African women may have it. While transplacental infection can occur, most infections happen during birth when the baby comes into contact with active sores in the birth canal.
And that's dangerous for the baby.
Extremely dangerous. Neonatal herpes can cause widespread infection, leading to brain damage, breathing problems, seizures, and has a high mortality rate, maybe 30–50% risk of transmission during an active outbreak at delivery if not managed properly, often with a C-section.
Okay, let's talk about substance use, starting with medications.
The golden rule here is crucial. Pregnant women should only take prescription medications if they are specifically prescribed by a doctor who knows they are pregnant. Many drugs can cross the placenta and harm the fetus.
What about over-the-counter drugs, things you buy at the pharmacy?
Those also need caution. Things like certain pain relievers, cold medicines, even herbal supplements should ideally be discussed with a doctor or pharmacist first. Some can pose risks, potentially causing birth defects, heart problems, or issues with blood flow in the baby, especially if taken frequently or at certain stages of pregnancy. Always best to err on the side of caution.
Now on to recreational substances. Nicotine from smoking tobacco.
The evidence here is overwhelming and unequivocal. Smoking during pregnancy is harmful.
What are the specific risks?
Increased risk of premature birth, miscarriage, stillbirth, low birth weight. Babies born to smokers are more likely to have poor lung function, visual problems, and are at higher risk for sudden infant death syndrome, or SIDs. There's even a condition called fetal nicotine syndrome, characterized by retarded growth.
Are there longer-term effects?
Yes. Prenatal nicotine exposure is linked to negative cognitive outcomes, learning difficulties, and behavioral problems later in childhood. Nicotine restricts oxygen flow to the fetus and is directly toxic. It also gets into breast milk, potentially causing restlessness and other issues in the nursing infant.
How big a problem is this?
It's significant. Some South African data is alarming, suggesting nearly 20% of babies might be born with nicotine levels indicating active maternal smoking, and another 30% exposed passively. And sadly, about half of pregnant smokers in SA continue to smoke throughout pregnancy.
Does passive smoking count?
Yes, passive smoking is also harmful. Even paternal smoking has been linked to negative outcomes, like potentially shortening daughters' reproductive lifespan.
Yeah.
The crucial point is, there is no safe level of nicotine consumption during pregnancy, and that includes e-cigarettes.
Okay. What about alcohol?
Alcohol is another major teratogen. Even moderate alcohol consumption during pregnancy can cause harm. The most severe outcome is fetal alcohol syndrome, or FAS.
What does FAS involve?
FAS is a cluster of problems, and it's irreversible. It includes cognitive deficits, often significant intellectual disability, slow physical growth, and a specific pattern of facial abnormalities, like small eye openings, a smooth area between the nose and upper lip, and a thin upper lip. Children with FAS also often have poor attention, hyperactivity, and sometimes cardiac defects. It's a leading preventable cause of intellectual disability.
And South Africa has a particular problem with FAS.
Tragically, yes. South Africa has the highest reported prevalence of FAS in the world. In some high-risk communities, rates might be as high as 5% or more. Given the potential for serious lifelong harm, the safest approach is complete abstinence from alcohol during pregnancy. There's no known safe amount.
What about illicit drugs?
Illicit drugs also pose significant risks. Drugs that cause physical dependence, like heroin or morphine, mean the baby can be born addicted and go through withdrawal after birth.
Withdrawal, what does that look like?
It's agonizing for the baby. Symptoms can include tremors, irritability, high-pitched crying, feeding difficulties, breathing problems, seizures, and in severe cases, even death.
And drugs that don't cause physical dependence, like marijuana or cocaine.
They are still harmful. Prenatal exposure to drugs like marijuana, cocaine, methamphetamine, or ecstasy is linked to a range of problems. Cognitive deficits, neurological issues, difficulties with emotional regulation, behavioral problems, and poor academic outcomes later on.
It must be hard sometimes to separate the drug effects from the circumstances.
That's true. Illicit drug use often occurs alongside other adverse factors like poverty, poor nutrition, stress, lack of prenatal care, which are also independently harmful to the fetus. However, even accounting for those factors, there's ample scientific evidence that these substances themselves have direct negative impacts. The message remains clear. Abstinence is crucial for the health of the baby.
Finally, what about the mother's emotional state? Can stress or anxiety actually harm the baby?
Yes. While we can dismiss old folktales about a mother's fright marking the baby, there is solid scientific evidence that extreme or, more importantly, prolonged stress, anxiety, or depression during pregnancy can have negative effects on the unborn child.
How does that work? What's the mechanism?
There seem to be a few pathways. First, when the mother is stressed, her body releases stress hormones like adrenaline and cortisol. These can cross the placenta.
And affect the baby.
Yes. They can reduce oxygen flow to the fetus, increase the fetal heart rate and activity level. And interestingly, the fetus's physiological stress response can last longer than the mother's own emotional feeling. Chronic exposure to high levels of these hormones can alter the developing brain systems involved in stress regulation.
Are there other ways stress impacts the pregnancy?
Yes. Severe stress can weaken the mother's immune system, making her more vulnerable to infections that could harm the fetus. Also, highly stressed pregnant women may be more likely to engage in unhealthy coping behaviors like smoking, drinking alcohol, or using drugs, which, as we've discussed, are direct teratogens.
So what are the potential consequences for the baby?
High levels of prenatal stress are associated with increased risks of spontaneous abortion, premature birth, low birth weight, and more difficult deliveries. Later on, it's linked to effects on brain development, leading to emotional problems, cognitive issues, language delays, and possibly conditions like ADHD. Some research even shows increased anxiety and depressive symptoms in offspring, detectable as early as age 2, linked specifically to maternal stress during pregnancy.
It really highlights that profound connection between mother and child right from the very beginning.
Absolutely. It underscores that the prenatal environment isn't just physical, it's biochemical and emotional too.
Okay, so after roughly 9 months of this incredible development, we arrive at the grand entrance, the birth process.
The crucial transition from the protective womb to the external world. And how this happens varies hugely across the globe, doesn't it?
Right, from very minimal intervention, maybe a home birth with a midwife, to highly medicalized hospital births, sometimes involving quite invasive procedures.
Let's start with the natural birth process, vaginal delivery. How does that typically unfold?
It usually happens in 3 stages. Stage 1 is the longest, involving the dilation and effacement thinning of the cervix. It needs to open up to about 10 centimeters.
How long does that take?
It varies hugely. Average is maybe 3 to 14 hours, but it could be minutes for some or over 30 hours for others, especially first-time mothers.
Okay, stage 1 is dilation. What's stage 2?
Stage 2 is the actual delivery of the baby. It starts when the cervix is fully dilated and ends when the baby is completely out. This stage usually lasts from about 15 minutes up to maybe 2 hours.
And stage 3?
Stage 3 is the delivery of the placenta, the membranes, and the umbilical cord, collectively known as the afterbirth. This usually happens quite quickly, within about 20 minutes after the baby is born, and it's generally painless. And first births tend to take longer overall.
Yes, typically. Subsequent births are often quicker.
Now, childbirth is often associated with pain. What about pain management? Medications?
That's often a big topic of discussion, even controversy. Various options exist, from narcotics or tranquilizers given early on to regional anesthesia like epidurals or spinal blocks that numb the lower body.
Were there concerns about these drugs affecting the baby?
Historically, yes. Since most pain medications do cross the placenta to some extent, there were worries about potential effects like drowsiness or breathing difficulties in the newborn.
What's the view now?
The modern perspective, based on extensive research, is that when these medications are administered responsibly by trained professionals, in appropriate doses, they generally have no significant long-term negative effects on the child's health or development. Of course, every woman's labor and pain experience is unique, so decisions about pain relief are very personal.
What about traditional medications used during childbirth?
That's a really important aspect, especially in contexts like sub-Saharan Africa. Estimates suggest up to 80% of the population there uses traditional medicine, and in South Africa, maybe 60–80%. Many traditional herbal remedies are used specifically during pregnancy and childbirth.
For what purposes?
Things like kagaba might be used to try and prevent labor problems, or preparations like crushed ostrich eggshell might be used to try and induce or stimulate labor.
Are these safe?
That's the major concern. While some traditional practices may be beneficial or harmless, others carry significant risks. There are reports of thousands of deaths annually linked to poisoning from certain traditional remedies used during pregnancy or labor. It really highlights a critical need for more research to identify which practices are safe and effective, and perhaps integrate them appropriately with modern healthcare, while discouraging harmful ones.
Besides vaginal birth, the other main option is cesarean section, or C-section.
Right. Delivery through a surgical incision in the mother's abdomen and uterus.
When is a C-section typically recommended?
Medically, it's recommended if vaginal delivery poses risks. For example, if labor isn't progressing despite interventions, if there are complications like the baby being in a breech, feet first position, if the baby is too large to pass through the pelvis, or if there's fetal distress or danger to the mother's health, like severe preeclampsia.
But sometimes women choose to have a C-section. Elective C-sections.
Yes, that happens too. Reasons vary. Some women may have intense fear of vaginal labor pain or potential injury. Others might worry about possible risks like brain damage or infection during vaginal birth, though these are rare. Sometimes it's about convenience preferring a planned delivery date.
Is there a financial aspect?
Yes, C-sections are generally significantly more expensive than vaginal births due to the surgical procedure, anesthesia, and longer hospital stays. In South Africa, this is reflected in the rates. C-section prevalence is much higher, around 75% among women with private medical insurance compared to about 25% in public hospitals.
What does the World Health Organization say about C-section rates?
Their guidance has evolved. Back in 1985, they suggested an ideal rate of 10–15% for any country, implying rates above that might be unnecessary. But by 2015, they shifted their stance. The focus now is more on ensuring that C-sections are available to all women who need them, rather than aiming for a specific target rate, acknowledging that needs vary based on population characteristics and healthcare access.
Are there downsides to C-sections?
Yes, while often life-saving, it is major abdominal surgery. Compared to vaginal births, C-sections are associated with slightly higher risks of both infant and maternal mortality, although the absolute risks are still low in well-resourced settings. Mothers also face higher risks of complications like infection, bleeding, blood clots, and require longer recovery times and hospital stays.
Does having a C-section affect the baby long-term or the bond?
Research suggests generally no. While some mothers might initially report feeling less positive about the birth experience compared to a vaginal delivery, studies haven't found significant long-term negative consequences for the baby's psychosocial functioning or the quality of mother-baby interaction due to having a C-section.
While most births go well, complications can happen, and the global picture is quite stark, isn't it?
It really is. It's sobering to remember that worldwide, almost 3 million babies die within the first 28 days of life each year. Another 3 million are stillborn, and around 300,000 women die annually from complications related to pregnancy and childbirth.
And most of these deaths are preventable.
Overwhelmingly so. Over 90% of these maternal and newborn deaths occur in developing countries, largely due to lack of access to skilled healthcare providers, basic facilities, and emergency obstetric care. It highlights massive global health inequities.
What are some specific birth complications? Anoxia.
Renoxia, or a lack of oxygen to the baby during labor or delivery, is a serious concern. It can damage brain cells. Causes might include problems with the umbilical cord, like it getting compressed, or the baby having breathing difficulties right after birth.
What are the effects?
It depends on the severity and duration of the oxygen deprivation. Brief or mild anoxia might have little long-term effects, especially with good postnatal care. But prolonged or severe anoxia can lead to brain damage, resulting in conditions like intellectual disability or cerebral palsy. Adequate prenatal care and monitoring during labor are key for prevention or early intervention.
Another common issue is low birth weight or premature birth.
Yes. Low birth weight, LBW, is typically defined as weighing less than 2.5 kilograms, about 5.5 pounds, at birth in developed countries. In developing countries, sometimes a lower threshold like 1.5 or 2 kilograms is used. South Africa often uses 2 kilograms.
And this includes babies born too early.
Right. LBW babies fall into two main groups: preterm infants, born before 37 completed weeks of gestation, and infants who are small for gestational age, SGA. SGA babies might be born full term but are underweight due to slow fetal growth, sometimes linked to genetic factors or placental problems.
How common is this in South Africa?
The rate is quite high. Around 14% of births are preterm or LBW, compared to about 7% in high-income countries. And there's a strong link to poverty, poor maternal nutrition, infections, and lack of access to quality prenatal care.
What are the risks for these babies?
Premature and LBW babies have a much higher risk of death in the newborn period. They're also more vulnerable to a range of developmental problems, health issues like respiratory problems, psychological difficulties, and intellectual challenges.
But the outlook isn't always bleak.
Not at all. This is where resilience really shines. Many premature and LBW babies, especially those who receive good medical care after birth and grow up in supportive, stimulating home environments, show remarkable catch-up growth and development. Often, by the time they reach school age, they display few, if any, lasting problems. It really speaks to the plasticity of development and the power of a nurturing environment.
That's really encouraging. Now, connecting all this back to the bigger picture, while birth is a universal biological event, how people perceive and experience it is deeply cultural, isn't it?
Absolutely. The biological processes are the same everywhere, but the meanings attached, the rituals performed, the support systems involved, these are incredibly diverse and shaped by cultural beliefs and values. And even within a single culture, you'll find variations, say, between urban and rural communities.
Can you give some examples of traditional beliefs, maybe around the placenta or umbilical cord?
Yes, those often hold great significance. For instance, the Maori people of New Zealand have a tradition of burying the placenta, whenua, in ancestral land, often planting a tree over it. This creates a deep, symbolic connection between the child and the land, also whenua.
Wow.
The Navajo people in the American Southwest also traditionally bury the umbilical cord within the sacred boundaries of their homeland to ensure the child remains connected to their people and heritage. In parts of Thailand, the placenta might be ritually cleaned, salted, placed in an earthenware jar, and buried under a tree chosen for its auspicious qualities. Hindu traditions also include various sacraments and rituals surrounding birth and early life, emphasizing purification and connection.
What about perspectives in sub-Saharan Africa?
Generally, across many cultures in the region, birth is seen as the ultimate expression and confirmation of womanhood. It often brings great status and joy. There can be a dichotomy, though. On one hand, it's viewed as natural, an achievement, a sacred event. Vaginal birth is often strongly preferred, sometimes seen as an important endurance test, proving a woman's strength.
But there's another side.
Yes, it can also be viewed with considerable fear and uncertainty as a potentially threatening or dangerous experience, given the historical and ongoing risks in many settings. So it's this complex mix of honor and achievement alongside potential danger and fear.
Are there any particularly unique cultural practices that stand out?
Oh, the Himba tribe in northern Namibia has one of the most beautiful traditions I've ever heard of. For the Himba, a child's birthday isn't counted from the day they are physically born.
No. When then?
It's counted from the day the child is first a thought in the mother's mind.
Seriously.
Yes. When a Himba woman decides she wants to have a child, she goes out alone into the wilderness. She sits and listens quietly until she hears the unique song of the child who wishes to come to her.
She hears the child's song.
That's the belief. Once she has heard and learned this song, she returns and teaches it to the father. They then sing this song together as they make love, inviting the child to join them.
That's incredible. Does it continue?
It does. While she is pregnant, she teaches the child's song to the midwives and the elder women of the village. When the child is born, these women gather around and sing the child's song to welcome them.
And after birth?
As the child grows, if they fall or get hurt, someone picks them up and sings their song to them. When they achieve something or go through rites of passage, the villagers sing their song to honor them. If they commit a crime, they are called to the center of the village, and the community sings their song to them, as a reminder of their true identity. It's sung at marriage, and at the end of life, the family sings it as they pass. It's this profound, lifelong affirmation of identity and belonging rooted before birth.
That is truly beautiful. It puts a whole different perspective on the beginning of life. Let's look closer to home, maybe some South African cultural practices. Traditional Xhosa culture.
Yes, in traditional Xhosa culture, childbirth is also a very important rite of passage, primarily managed by women. The labor typically takes place in a separate hut, a rondavel, attended by experienced elderly women, often referred to as grandmothers. Men are usually excluded.
Is there a period of seclusion after birth?
Yes, the mother and newborn remain secluded, often until the baby's umbilical cord stump falls off. This process might be aided by applying a traditional paste, perhaps made of ash, sugar, and the juice of a plant called umtuma.
Are there specific rituals during or after this time?
There are several. Around the third day, the sifudu ritual might be performed. The baby is gently passed or floated over pungent smoke from smoldering sifudu leaves, which is believed to instill courage and prevent shyness later in life. The mother might also pass the baby under her bent knees to impart strength. The baby is washed and often smeared with white clay, inkuk, or powder from the sweet-smelling ntombori wood, seen as protective and cleansing.
What about the umbilical cord and placenta?
The Inkaba ritual is very significant. It involves the careful burial of the dried umbilical cord stump and sometimes the placenta, often near the family home or at a significant ancestral site. This act is believed to permanently seal the child's spiritual connection to their ancestral land and lineage.
And then the child is introduced to the community.
Yes, usually around the 10th day, the umbilical ceremony takes place. This is a major event where an animal, typically a goat, is slaughtered as an offering to the ancestors. There's a feast, and the child is formally named and welcomed into the family and community. A strip of the goat's hide might be worn by the baby as a symbolic connection and protection.
Fascinating. And Zulu culture. Similarities or differences?
Some similarities, some differences. Childbirth in traditional Zulu culture is also strictly a women's affair, with older women acting as midwives. Men are typically not allowed near during the birth.
What happens immediately after birth?
The baby is usually washed, sometimes in the umsamo, a sacred part of the hut, often with intalizi, which are protective herbal medicines mixed with water.
Is there an isolation period here too?
Yes. The mother and child are typically isolated, often until the umbilical cord falls off. During this time, the mother might be considered ritually unclean, okusla, and may use special utensils and avoid certain activities. This seclusion usually lasts about 5 to 10 days.
And after the isolation.
A purification ceremony is held for the mother. It's often only after this ceremony that the husband is allowed to see the child for the first time. Traditional beer, ochwala, is usually brewed for a celebration to thank the ancestors and the midwives and formally welcome the baby.
It's just incredible. What an astonishing journey we've covered. From that microscopic union of cells through the intricate dance of genetics and development.
Navigating the potential challenges, the different ways birth unfolds, and finally seeing how culture weaves such a rich tapestry around this universal human experience.
Yeah, it truly is an astonishing process.
Hopefully you now have maybe a deeper appreciation for the sheer complexity and resilience involved in human development before birth.
It's certainly a powerful reminder, isn't it? That every single life begins with this incredibly intricate interplay of biology, environment, and sometimes, as we saw, profound ethical choices.
Choices that ripple outwards, affecting individuals, families, and society as a whole.
So as we wrap up, maybe a final thought to leave with our listeners. Considering the profound, lifelong impact of these very earliest moments, what responsibility do we have as a society? A responsibility to ensure that every first chapter, every prenatal journey, is given the best possible chance to unfold well, regardless of the circumstances surrounding it. What does being truly well-informed mean when it comes to supporting new life, and how can that knowledge actually translate into meaningful support and action?